Hello students, I am Dr. Neelam Mahajan. I shall be discussing Parenteral Products Part 2 Production today. In this module, we will introduce ourselves to production of parenteral products, packaging of parenteral products, stability of parenteral products, and quality control tests of parenteral products. Production process of parenterals comprises of steps ranging from collection and combining of formula ingredients to addition of the product in individual containers for distribution. Personnel carrying out these steps and facilities in which they are performed have close association with these steps. Effectiveness of an ideally planned process can be increased with right attitude or training of personnel involved or provision of efficiently controlled environmental facilities concerned. Assured success of manufacturing operations is possible through carefully written process steps after their effectiveness is established. These written process steps are generally called standard operating procedures or SOPs. Extemporaneous changes in these procedures are not allowed. All changes go through approval steps similar to originally written SOPs. Further, as per FDA's good manufacturing practices, at the end of the production process, it should be assured that all steps have been performed as prescribed. This is even more significant than those from product release testing. This requires extensive record keeping. Such in-process control is essential for assured product quality. Production of a quality product is the result of the continuous dedicated effort of the quality assurance, production and quality control team in development of and adherence to effective SOPs. Quality assurance is preventative development process. It differs from quality control, usually involving pre-planning factors affecting quality of a product. Quality control may include QA in case there is only one organizational group directly responsible for quality in a plant, but it chiefly includes operations and tests designed to evaluate actually achieved product quality. Let us discuss the passage of materials through various steps of production process. In the initial step, formula ingredients, container components and processing equipment released for use are taken out from their respective storage areas. Process equipment and container components are cleaned thoroughly as per required specifications, are assembled in a clean environment and preferably are sterilized and depyrogenated prior to use. Ingredients are compounded according to the master formula in an environment in which a high level of cleanliness is maintained. A solution product is filtered during transfer to aseptic filling room. All equipment and supplies introduced into the aseptic filling area are sterile and come directly from sterilization process. If not possible, packages, hose lines from equipment and supplies are passed through ports of nominal size so as to allow prompt reclosure under aseptic conditions. Outer wrappings of packages are loosened and inner wrapping is grabbed by personnel in the aseptic filling room. Outer surfaces of packages, boxes or equipment are wiped with a disinfectant solution while transferring them into aseptic room. All supplies are introduced into aseptic filling rooms in a manner so as to maintain their aseptic conditions. This prevents environmental contamination of product during its redistribution into individual containers. After sealing these containers, contamination of the product is not possible. Product is sealed in its final container within aseptic room. It is then transferred to the packaging area. Packaging area is maintained clean but need not meet the standards compulsory for aseptic rooms or for compounding room. Packaged products are placed in quarantine storage until the completion of all tests and evaluation of in-process control records. then the product may be released for distribution. Now talking about the packaging of parenteral products. Package is an extremely important part of the product. It presents the product to the user. It must be particularly distinguished, tidy and eye-catching in appearance. Also, it should reflect quality, purity and dependability of the product, as well as conformance to QA and QC principles and procedures as an inherent part of production of product. Package also must provide accurate and complete information essential for its use. Its label should be readable with clearly evident identity and strength of the drug. Furthermore, package should protect the product against physical damage during shipping, handling, storage, and the package should also protect light sensitive substances from ultraviolet radiation. Further, packaging requirements for injections as per USP will be discussed. Volume of an injection in single dose containers should not be more than 1 liter and should provide amount specified for one-time administration. This minimizes likelihood of use of remaining residue at a later time. Preparations intended for intraspinal, intracisternal or peridural administration should be packaged only in single dose containers because of the sensitivity of nerve tissue to irritation from added substances like antibacterial agents. Normally, multiple dose containers shall not contain a volume of injection more than 30 ml. Injections labeled for veterinary use are exempt from above limitation to single dose containers and volume of multiple dose containers. When a label is put on a container, a sufficient area of container is left uncovered to permit inspection of the contents. These packaging and labeling stipulations are basic guidelines for all products. Operation of packaging department for sterile products is essentially similar to that for other pharmaceutical packaging departments. Chief objective is proper labeling and packaging of all units with adequate controls for its assured accomplishment. Now we will discuss the stability of the product. Packaging components are in intimate contact with the product, so it is necessary to evaluate effect of all components on the stability of the product, particularly when product is subjected to accelerating reactivity of thermal sterilization. Now, quality control. Three general areas of quality control include incoming stock, manufacturing and finished product. First of all, we will discuss quality control with respect to incoming stock. For sterile products, incoming stock control includes routine tests on all ingredients as well as special evaluations like PYROGEN tests, then glass testing on containers and identity testing on rubber closures. It may also be necessary to perform microbial load test to determine the number and types of microorganisms present. Process control area in the manufacture of sterile products involves all of the innumerable number of tests, readings and observations made throughout the manufacturing process of a product, which include conductivity measurements, then confirmation of volume of fill in product containers, recording of cycle time and temperature for thermal sterilization of product, and confirmation of the count and identity of labels for the product. Now, production control. Production control includes all final tests and assays of product, namely routine chemical and biological tests, a leak test, a clarity test, a PYROGEN test and a sterility test. First of all, we will discuss leak test. Now let's talk about significance of leak test. Ampules are intended to provide a hermetically sealed container for a single dose of a product. Likelihood of incomplete sealing of tip sealed ampules is more than those which are pull sealed. Due to improper handling, small cracks may appear around seal or at the base of the ampule. These capillary pores or tiny cracks present in ampules serve as entrance points for contaminants, may lead to leakage of contents and may spoil the look of the package. Expansion and contraction of ampule and contents induced by temperature changes during storage facilitate interchange between contents of sealed ampule and its environment if an opening is there. Leak test is meant to detect and discard incompletely sealed ampules. After having learned the significance of leak detection test, we will discuss the method of leak detection. Usually, leaks are detected as per a standard procedure in which a negative pressure is produced within an incompletely sealed ampule with ampule entirely sunken in an intensely colored dye solution, preferably methylene blue. Due to subsequent atmospheric pressure, dye penetrates an opening, if any, which is visible after external washing of the ampule. Vacuum is sharply released after half an hour. Only a tiny drop of dye penetrates a small opening. More effective detection of leakers is done by immersing ampules in a bath of dye during autoclaving. The method of leak detection has certain limitations. Capillaries of about 15 micrometer in diameter or smaller may or may not be detected by these methods of leak detection. Vials and bottles are not subjected to such a leak test because rubber closure is not rigid. However, bottles are often sealed while a vacuum is being pulled so that the bottle remains evacuated during its shelf life. Presence of vacuum is detected by striking the bottle base sharply with the heel of hand or by applying a spark tester probe to outside of the bottle, moving from liquid layer into the air space. Now we will discuss clarity test. This test is also very significant. As we know, clarity is a relative term. Its meaning depends on subjective evaluation of the observer. A clean solution with a high polish is indicative of exceptional product quality and purity. Preparation of sterile products perfectly free from visible particulate matter is practically impossible. Consequently, it is the responsibility of the quality control department to detect and discard individual containers of a product to be considered unclean by the ultimate user. As per USP, good pharmaceutical practice requires visual inspection of all containers and discarding those with visible particles. Parenteral solutions can satisfy the standards which we just now discussed. Further, product development, raw material quality control and process control have led to elimination of all possibilities of widespread particulate development. However, a few containers with visible particles detectable by visual inspection should be discarded. Although particulate matter is of primary concern in intravenously given products, all parenterals should be free from insoluble particles and should be exceptionally clean. Suspensions, emulsions or dry solids and solutions should be compounded and processed under clean conditions to minimize presence of foreign particles. Now we will discuss the methodology of clarity test. Visual inspection of a product container is usually done in this test. It involves individual human inspection of each externally clean container under good light, baffled against reflection into the light, and viewing of the container against a white and black background. The contents of container are set in motion with a swirling action to facilitate visibility, but introduction of air bubbles should be avoided. Then comes the step of inverting the container so as to see heavy particles. It is the final step in inspections. Human visual inspectors' efficiency reduction due to eye strain, fatigue, distractions and emotional disturbances is possible. However, human visual inspection offers the advantage of inspection of 100% products with a level of discrimination at par with that of the user. Instrumental methods based on principles of light scattering, light absorption and electrical resistance are used for getting particle counts and size distribution of particulate matter in liquids. In all methods, however, product unit is destroyed to take test sample. Thus, these are used only for quality control testing. A method using video image projection coupled with electronic circuitry detects moving particles without destructing product unit. This can be used for inline detection of particles in product units. Now we will talk about pyrogen testing. Presence of pyrogenic substances in parenterals is determined by a qualitative biological test based on fever response of rabbits. Rabbits are used as test animals because they share the same physiological response to pyrogens with human beings. On injecting a pyrogenic substance into the rabbit vein, temperature rise is observed within a period of 3 hours. Housing conditions and handling are critical to obtaining consistent results with rabbits in the test. Rectal thermocouples remain in place throughout the test, hence these are used in place of rectal thermometers. No rabbit handling for individual temperature readings is required. 100 or more animals a day can be handled as compared to 15 by individual thermometer method. Many medical agents with antipyretic or other interfering effects, if present, interfere with test results. Pyrogen test is performed on all vehicles used for injections, but only on finished products that do not interfere with the test, have a high tendency for contamination with pyrogens, or are given in large quantities. Considerably greater danger exists from injections of large volume solutions containing pyrogens than from smaller volumes. Also, the pyrogenic effect is less with intramuscular injections as compared to intravenous injections. An in vitro test method for pyrogens utilizes gelling property of lysate of the amoebocytes of Limulus polyphemus. Now it's a turn to talk about sterility test. First of all, we will discuss the significance of conducting sterility test. All products labeled sterile must pass sterility test after being subjected to an effective sterilization process. Test for sterility is meant for detection of viable microbes in official preparations. Now coming to its methodology, sterility testing of the products is carried out either by membrane filtration method, also called as method A, or by direct inoculation method called as method B. Membrane filtration is used for filterable aqueous preparations, alcoholic or oily preparations, and preparations miscible with or soluble in aqueous or oily solvents, provided these solvents do not have an antimicrobial effect in test conditions. Test for sterility is carried out aseptically while taking care to avoid contamination. Working conditions in which tests are conducted are monitored regularly by appropriate sampling of the working area and by carrying out appropriate controls. Now we will talk about media for sterility testing. Sterility test media should support growth of aerobic microorganisms, anaerobic microorganisms, and fungi. Fluid thioglycolate medium is chiefly intended for culture of anaerobic bacteria. However, it also supports the growth of aerobic bacteria. Soybean casein digest medium is suitable for the culture of both fungi and aerobic bacteria. Fluid thioglycolate medium is used for clear fluid products, and for turbid and viscid products alternative thioglycolate medium is used. Fluid thioglycolate medium devoid of agar and risazurine is alternative thioglycolate medium. Now we will discuss method A of sterility testing by membrane filtration. This method makes use of a suitable unit consisting of a closed reservoir and a holder. Between these two, a properly supported porous membrane is placed. A membrane suitable for sterility testing should have a nominal pore size with well-known effectiveness to retain microbes. For example, cellulose nitrate filters for aqueous, oily, and weakly alcoholic solutions, cellulose acetate filters for strongly alcoholic solutions, and specially adapted filters for certain products like antibiotics. Before sterility testing, membrane is rinsed with diluting or rinsing fluid. For aqueous solutions or Soluble solids, membrane is rinsed with sterile solution such as fluid A comprising of 1 gram of peptic digest of animal tissue in 1 liter water. For testing oil, oily solutions, ointment and water in oil type emulsions, membrane is rinsed thrice. About 100 ml of a suitable sterile solution with a suitable emulsifier like polysorbate 80 at a concentration of 10 gram per liter is filtered through it each time. Addition of surfactant makes it useful for formulations having lecithin or oil. For products with antimicrobial properties, membrane is washed at least thrice. Each time, volume of the chosen sterile diluents is filtered through it. Rinsing should never exceed a washing cycle of 5 times 200 ml, even if it does not fully eliminate antimicrobial activity. After rinsing membrane, contents of the container to be tested are transferred to membrane. Aqueous and oily solutions of sufficiently low viscosity may be filtered without dilution through a membrane. Viscous oils, ointments in a fatty base, and emulsions of water in oil type may be diluted as needed with a suitable sterile diluent like isopropyl myristate. Fluid products in pressurized aerosol form are frozen in an alcohol dry ice mixture at least at -20°C and added to fluid D before transfer of contents. In case of pre-filled syringes without attached sterile needles, contents of each syringe are expelled into 1 or 2 separate membrane filtered funnels or into separate pooling vessels before transfer. If a separate sterile needle is attached, syringe contents are directly expelled. After the contents are filtered, membrane is aseptically cut into 2 equal parts. Each half part is transferred to culture media. Fluid thioglycolate medium is incubated at 32.5 plus minus 2.5 degrees Celsius, whereas soybean casein digest medium is incubated at 22.5 plus minus 2.5 degrees Celsius for not less than 14 days. These media are examined for macroscopic evidence of microbial growth at intervals during and at the end of the incubation period. If no evidence of microbial growth is found, product complies with the test for sterility. If there is evidence of microbial growth in the form of development of bacterial colonies, product under examination does not comply with test for sterility unless test is invalid for causes unrelated to the product to be examined. Test may be considered invalid only if one or more of the following conditions are fulfilled. The conditions are: the data of microbiological monitoring or sterility testing facility or the test method used during test shows a fault, and negative controls also show microbial growth. If the test is declared to be invalid, it is repeated with similar number of units as in the original test. If no evidence of microbial growth is found in repeat test, product tested complies with the test for sterility. If microbial growth is found in repeat test, the product tested does not comply with the test for sterility. Now we will talk about method B, that is direct inoculation method for sterility testing. Direct inoculation method is utilized for testing oily solutions, creams, ointments, solid products, surgical devices, sterile devices, sutures, surgical dressings in cases where membrane filtration method appears difficult. In direct inoculation test, quantity of the preparation to be examined is transferred directly into the culture medium. Volume of product should not be more than 10% of the volume of the medium unless otherwise prescribed. Oily liquids, creams and ointments are diluted to about 1 in 10 by fluid D. Sterile devices can be plunged intact into selected medium. In case of surgical dressing, 2 or more portions from innermost part are aseptically removed and immersed intact in unit. In case of catheters, where inside lumen and outside are required to be sterile, they are either cut into pieces so that the medium is in contact with the entire lumen or fill the lumen with the medium. If product to be tested has antimicrobial activity, the test is carried out after neutralizing it with a suitable neutralizing substance or by diluting it in a sufficient quantity of culture medium. For large volume of product, it is preferable to use a concentrated culture medium prepared for subsequent dilution. When appropriate, concentrated medium is directly added to the product in its container. Inoculated media is then incubated for not less than 14 days. Cultures are observed several times during incubation period. When thioglycolate medium is used for detecting anaerobic microbes, shaking or mixing should be kept to a minimum to maintain anaerobic conditions. If material being tested makes medium turbid and presence or absence of microbial growth cannot be readily determined by visual examination, 14 days after beginning of incubation, transfer portions of medium to fresh vessels of the same medium and then incubate the original and transfer vessels. Results are interpreted as in membrane filtration method. So students, in this module, we discussed and learnt about various aspects of production of parenteral products. We learned that production of a quality parenteral product is the result of continuous dedicated effort of quality assurance, production and quality control department. We learned that quality assurance is a preventive development process for a quality product, and quality control includes operations and tests designed to evaluate the actually achieved product quality. In nutshell, we discuss the usual steps of production process, requirements of packaging for parenteral products, stability of the products, various quality control operations and tests designed to evaluate actually achieved quality of the parenteral products. Thank you.